recording. All right, cool. So the reason why I wanted to talk about interview perspectives, interview-based style answering for signal management is recently we had several students who have applied for jobs related to safety science, related to safety physician roles, some titles based on that. And in their job descriptions, I have seen most of the companies have asked for safety surveillance skills, a safety scientist role or safety physician role, uh, more towards safety profiling. So when I say safety profiling, it not only involves signal management, you need to make sure you understand the benefit risk profiling of the drug or the safety profiling of the drug, where you are keeping a very close, keen look on the signals along with the aggregate report submitter are going to be submitted, and the risk evaluation plans that are in place are going to be implemented if your drug is post marketed. So just for the clarity sake, let me put it this way. When you are talking about safety surveillance, you are mostly talking about post marketing. You're not talking about clinical trial drug. Because there's no surveillance for the small denominator of patients exposed in your clinical trial. So all the time, safety surveillance always, like, by default goes for post marketing. But sometimes we kind of interchangeably use it for, clinical trial products as well. So when you hear the word safety profiling, what do they mean is by this, by your knowledge, by your expertise in signaling risk, signal workup, making sure you can characterize the risk and evaluate a good mitigation strategy and implement the mitigation strategy for that risk, along with evaluating it, re-evaluating it, and re-implementing it. So risk management is like a cycle. So your ability to do that, along with how are you going to summarize your findings from signaling and as well as risk evaluation plans into your ongoing aggregate reports, especially if it is a PADER for FDA or PBER for other regions like EU. How are you going to summarize those findings? Ultimately summarizing all those things into your safety sections of that aggregate report, where you go into benefit risk statements. Is it favorable? Is it not favorable? What are you doing to make it favorable? All those things would happen in the benefit risk analysis sections, where as a safety physician or a safety scientist, you need to write that concluding statements. So from that perspective, we were doing a lot of I was doing a lot of interview preparations, and I just wanted to go over certain things from a signal standpoint, because even though our students have gone through signal management, they kind of seem to be not clear about what are they talking about. Is this a clinical trial product or a post marketing product per se, based on SIAM's 5 guidelines, or SIAM's, I believe SIAM's 8, sorry, SIAM's 8 guidelines. There is a book called uh, Practical Aspects in Detection, Signal Detection, or something on that, pragmatic approaches to signal management. So that's on SIAM's 8. It's a very good reference book that you should read for signal management. And what the summary of that book that I have to kind of reflect on here is, the signal management is mostly implies to a post marketing drug. When you hear signal management, when you hear the steps of signal management, you always think about a post marketed drug. But for a clinical trial product, you can use the same steps, you can use the same, I not say steps, you can use the same methodology in signaling. So we call it signaling because it's not actually signal management. Because for signal management, let us say you find a signal, you may have to do some recommended action steps, change the label, change the RMP, change the PADER, change the PBER, report it to health authorities. There are so many regulatory actions you may take for post-marketing drug, because the public health may be impacted because of the signal that you found out. But whereas when it comes to a clinical trial product, you really don't have a label, you don't have an RMP, you don't have any kind of ongoing aggregate reports other than DSUR, but it is good to have a signal identified and mitigated, because your patients that are enrolled, your subjects enrolled in clinical trial will be impacted because of that signal. So most of the time, signaling is very good in phase 3 studies. Even though the word the word signal is used, it's only for post marketing. The methodology in which you will uh, get that question first, identify the signal, and how do you work up a signal by the resources you have within the company, and then comparing it with other comparators and going into larger databases. All those methodology is the same, but the recommended steps for that clinical trial product signaling is very different, and you will probably end up mitigating the signal. If I have to use the word signal for clinical trial product, you mitigate it by changing the protocol. You will add something in the protocol to protect the patients. Let us say, for example, you found out that your drug is in phase 3, and you had some reports of cardiac toxicity in phase 1, and but it was not severe, so that you went to phase 2. Phase 2, you still had some cardiac tox. Say for example, you had some ECG abnormalities, very minor, like phase 1, I mean, maybe first degree AV block, not like a QR prolongations or QT prolongations. So you had first degree AV block, but still you had some electric imbalances in your patients. But phase 3, you found out, because you increased the exposed number of subjects in phase 3, you found a little more increased severe electrical imbalances, which mandated you to work up this signal. So in that case, if you found the cause, I mean, the signal is all about the cause, the strength of evidence between the drug and the event combination, drug event combination, how strongly they are related is the signal. So if you find a very potential, very good strength between the drug event combination, you want to protect your patients. How do you protect your patients? Well, that will be enrolled in the future is by saying that, look, every time, even before the subject is enrolled in your clinical trial, probably you will do an EKG to make sure their electrical cycle is normal. They have a normal EKG before they get enrolled in the trial. Maybe you're going to have a cardiology consultation before they enroll in the trial. Maybe once they enroll in the trial, before administration of the drug, you want to make sure that they have a normal EKG, and they are actually, the drug is administered in an ICU setting, so that if there is any kind of cardiac toxicity or any electric imbalance, it can be corrected. There should be a defibrillator, or some medical management has to be done right away in an intense setting. So maybe your drug should be given, administered most likely IV, only in an ICU setting, so that it can be any kind of untowards fatal or life-threatening events can be avoided. So you can mitigate that cardiac toxicity because you found the strength by signaling, by changing the protocol. And maybe you're going to have, I mean, for sure, if you change the protocol, you'll get a protocol amendment. It will go into the IB next year. You will have this signal going into ongoing signals section of a DSUR. That's the maximum you do for a clinical trial report or a clinical trial signaling. But for post marketing, there might be a lot of implications, that, as I was alluding earlier. So that is the reason why I want to clarify between signaling that we do for clinical trial products versus signal management for post marketing. And people use these words very loosely, but we want to be careful when we say signaling. We are just using the same methodology of a post marketing signal management for clinical trials. By the end of the day, if you don't remember all these things that I just explained, understand this: patient's health is of your primary importance. So if you are doing anything for patient health, whether it is post marketing or in clinical trials, if you can mitigate the risk by all your means, that is more than sufficient for your drug to prevail in the market or drug to go through clinical trial products. So the patient safety is important. So that's number one. Number two of three is students are asking about, what kind of examples can we give for signaling. So, guys, don't complicate it. You don't need to come up with earth shattering examples. No, I have not found a signal where the drug was stopped in clinical trials in my 18 years of PV experience. So, you are not going to find a signal where, oh, I found a signal, let us stop $1 billion drug from developing. That will never happen. It's not practical. So, when you kind of want to give examples in the interview process, just pick up some examples that you can relate to. Like for example, recently for AstraZeneca COVID vaccine, they found out that this vaccine can produce paralysis, can give you stroke, can lead to thromboembolic phenomenon. So it's a very potential risk. So what they did is they are not giving this they're not giving this COVID vaccine to certain class of people who are more susceptible to those kind of stroke episodes. So again, very simple examples. You don't need to give something very profound. I always give same examples in our modules because it's easier to follow. For example, let's say your drug is metabolized in liver, excreted through urine. You're working on oncology drug, which is pretty much the mechanism of how the pharmacokinetics work in the body. For that drug, if the body is exposed to the drug in like 6 cycles or 12 cycles, there is a risk for you to get into hepatic failure because the drug is going through liver all the time, pushing their LFTs up. AST, ALT, GGT, bilirubin, all those things. We can expect the ALTs to be elevated, but not to a point that the elevated LFTs leads to Hy's law. So if you are going for any of the safety scientist or physician roles, look up for Hy's law, HYS law. So you need to read about it. What is Hy's law? How do you mitigate it? What is the definition of Hy's law and all those things. So it should not go to the point of having an Hy's law case, because if you have an Hy's law case, probably you have to adjudicate the case. You need to go deeper into that one single case and see what confounders you have. You might have to have a data monitoring committee, which is a third biased committee looking into that patient, even one case of ISLA, because it's completely preventable. You can prevent the death of a patient because you looked into it. So that's why we don't really want a ISLA, but it happens all the time. I adjudicated several ISLA cases for several oncology products. So you need to make sure that you are doing this signaling signal workup for that elevated LFTs. So how do you do that in a clinical trial product is, you have to have that question mark. See, the point is identifying the signal is always at the hands of the physician, at the hands of the safety scientist, probably looking at the data or a single case. I mean, one single case, until unless it's a sentinel case, you will not say it's a signal. But you need to have a keen eye on the adverse event reported. Let's say you need to know your preclinical data, you need to know your clinical trial data, you need to know your post-marketing data if it is available, and then see that, okay, what is this adverse event reported? In which system organ class is it? Are there any other toxicities in that system organ class? Is this something related from a pathophysiology standpoint? Is this something related to what we already know? All these questions have to run in your head when you are looking at that case and doing medical review of that case. And now, once you do medical review, yes, at a case level, at that one subject level, you might say it is not related because that patient had some confounders. Let us say the same LFT, AST, ALT were 3 times the higher limit, but this patient had previous episode of, let us say, cirrhosis, or the patient had previous episodes of hepatitis, or an ongoing hepatitis C, chronic hepatitis. All these things are confounders, clear confounders for that patient to have elevated LFTs. So you can say it is not related in the presence of very strong confounders, but that should trigger the signal workup. Like, look, this patient, yes, I understand, is not related, but out of all the patients so far, can you dig up the data and see how many patients had LFTs increased? Is there any confounders? That is signaling. Someone doing that workup for you is signaling in clinical trials. So if you give that example of elevated LFTs and You don't need to run the whole process. I was just talking to the student for the interview, and she was asking me, oh, so what kind of recommended steps do you do for that signal? You don't need to go that far. You can just say that we identified a signal of elevated LFTs. We did our workup from the data, the information that we have from enrolled patients within the database we have. Probably did some um, data review, and we found out that only 10% of our patients exposed had grade 2 LFTs, and we want to monitor the signal. You're not refuting it, you're not validating it, you're not doing any recommended regulatory actions, but you're continuing to monitor it. The reason is they are just grade 2 LFTs, which doesn't have any clinical significance. They're non-clinical significant adverse events. Is it clear? So this is the second thing I want to tell you. Don't try to, kind of be the star in the interview by giving some examples that you cannot justify yourself. So try to do some minimal thinking, elevated LFTs. Let us say for oncology, immunology products, you can start off with cytokine release syndrome, something very mild, which doesn't involve any kind of fatal life-threatening conditions, and say that we were monitoring the signal. We had a signal log and we are monitoring the signal. So that is pretty much what I wanted to tell you. Is that clear, guys? Yes, yes, no.
Yes.
Is it too much?
Yes, doc.
No, not too much.
All right. The third and the last thing, before I open up for any questions on this, is from an interview standpoint. Again, very straightforward. All your signals that you are working on, whether in whether in clinical trial or in post marketing, you are not the only person doing it. Let me put it that way. You're not the only person doing it. There will be a lot of other people who will be supporting you. There'll be a whole safety team, signal evaluation committee helping you, but you will have your role to play. As a physician, you will have your role to play. As a safety scientist, you'll have a role to play. And normally, if the signal is really strong and it requires some regulatory actions, many people will be involved. Definitely, your functional head of your safety will be involved. And they will go into cross-functional teams. They will present this signal, and then it goes to clinical, it goes to medical affairs. So there will be a very large team once you identify and validate, and if it mandates a regulatory action, it's not something you do overnight. It will take months to do it, in even to communicate to health authorities. And again, it depends on the severity of the signal. If you find some emerging safety information, you have three calendar days, according to EU. So it's not like you take your own sweet time. You got to communicate it faster. But still, most signals, if they're not severe, doesn't have public health impact, you may take 3 to 6 months to actually communicate those signals to the concerned people and escalate it to a point that as a pharmaceutical company, you may have to communicate it with health authorities. Pretty much, say, hey, FDA, this is what we found. We are working up on the signal. We'll give you further updates, so on and so forth. Not that every SUSAR, or sorry, every report in post marketing FDA is reading it. So no one reads all the cases that FDA receives. I believe FDA is receiving thousands of cases every day, and no one is actually manually reading it. But when your product is being evaluated, whether it's post marketing or in clinical trial, for sure they're going to go through those cases. So you don't want to catch any surprises at that point. So that's why you have dear doctor letters to the PIs, you have dear doctor letters to the prescribers, you will actually have this dear investigator letters. So you'll do all kinds of communications to regulatory authorities, to PIs, to prescribers, all those things you'll kind of do. So the point to be noted is, students, when they go into interview and when they talk about signaling, they seem to be thinking that I'm the only person in signaling and I'll be doing everything A to Z. That's not how it is done. You'll have a team to do it. Am I making sense? So these are the three things I just want to tell you from an interview standpoint. because these are some of the questions that I answered in the recent interview preps. So I thought it would be helpful for you. Is it valuable? Is it something that you understand, guys? Is it too much?
Yes.
Yes, doc.
Thank you.
Okay.
It's good.
All right. Any questions on this? Any questions on these three elements that we just went over?
I do have one question, doc.
Sure. Go ahead. Be louder.
So, yeah, so with the Hy's law case, if it happens in the clinical trial, what happens?
No, one case of ISLA, where the patient met with some fatal or life-threatening situation going into hepatic failure because of your drug, then you have to adjudicate it. You have to make sure it doesn't re-happen again.
Okay. In case of post-marketing, if it happens?
The same thing, the same thing.
Okay. So one
The
case
point
doesn't make.
is, the point is, how is the life of one patient in clinical trial different from one patient in
No, no,
post-marketing?
not one patient in the post-marketing case. Minimum of how many cases would make it much worse?
No, no, it's not how many cases. Even if you have one case of a fatal HISLA, the question is, what did you do to mitigate it to the second one?
Okay.
So that is the question. It's not like how many how many victims have to be found to make sure you
No, no,
clear
because of the
the risk.
frequency in the, no, my question is because of the frequency and the strength of evidence increasing, does it make any changes?
No, no, no. See, those are all signs, but from a drug development from a pharmaceutical standpoint, your science doesn't have any impact. If the health of a patient is deteriorating because of your drug, you better take action yesterday. Yeah, all the things that you are learning is all good on paper, but when it comes to patient's health, what did you do to mitigate it? Yes, you came to know about Hy's law last 15th of July, and now a patient is taking the same drug on 22nd of 24th of July. What did you do in this 10 days is the question.
Okay. Thank you, doc.
So Hy's law doesn't really, it's not like Hy's law is detrimental or bad to the program, to the clinical developmental program. It's not like that. But did you take your due diligence action? So for one of the thing would be, before you were not checking for LFTs before administration of the drug. That's why this Hy's law happened, let us say. Now you're going to mitigate it by saying that before you are administering the drug, we need to see normal LFTs, or we need to see LFTs are below the second higher limit. So some restriction has to be placed so that it is not bumping up. Am I making sense?
Yes, doc.
And sometimes we give this hepatoprotective drugs to mitigate it. Sometimes we give steroids, steroids in a tapering dose, so that it kind of smoothens the effect. So we do a lot of things to mitigate it, but it's not like Hy's law is equal to death of the clinical developmental program. That's not how it works. How many of you know Hy's law? What is Hy's law? Okay, thanks.
The
If people are smiling, I know you do not know. So check out Hy's law. It has three
it
criteria for Hy's law. Go ahead, Sri Lakshmi.
the one I read was like it has both bilirubin as well as the ALT and AST increase in it in the patients.
So it is like you have a clear definition, guys. It's not that vague. It's a very clear definition. The bilirubin has to be twice of higher limit, and the AST/ALT has to be three times of higher limit, leading to hepatic failure, leading to the death of the patient. So this is the definition, Hy's law. You don't need to read up a lot. Just to read what is in Wikipedia for Hy's law. Just read Wikipedia. That's what I did when I did not know Hy's law. And then you can read a book on it. There is a Siam's book on total drug-induced hepatic functions. There's a book on it, almost 60-page book. If you want to buy it, if you want to download it on internet, you can do it. I think it's free right now. When I studied it, I had to buy it, but now they're all free. You can download the PDF, read through the 60 pages. You will cover everything that is drug induced for hepatobiliary system, all the drugs, everything that you need to know. The same way for cardiac toxicity, SIAMS has a workshop. You can download that book, read through all the cardiac toxicities and how you mitigate it. So for every major system organ class which has some drug induced effects, you have CIOMS guidelines. So you got to read about Hy's law. So if you guys come to interview prep and if you're going to like be where you are, you're going to have a very rough time and you're going to miss on interviews. I don't want you guys to do that. Just prepare more right now. This is the time you need to prepare on all those things. All right, guys. Any other questions? No. So again, I believe as I was as we started, these are all some of the things that I've been going through with students in interview prep, and that's why I want to share with you. Again, we are going to meet in 2 weeks' time back here. We are missing probably 3, 4 folks. If you kind of know them, just let them know these calls are very important. This is not a content that will be delivered every time, and sometimes these recordings also don't make it up into your LMS sometimes. So it's always good to be in the class for 30, 40 minutes, get to know this. And I make a notes out of what I'm doing on a regular basis, like how I did for this class around, so that we can discuss more. And most important is you have until 15th of August to complete the signal management. So try to go over it. You may need to go over it 2, 3 times to kind of understand it from a practical standpoint. So just to do your work right now so it becomes easier later on. So when we meet in 2 weeks, we are going to open up risk management. I'll take it slow with risk management. We'll do maybe 4, 5 topics at a time so it becomes easier for you because you're still lagging behind our signal management. I'll try to break it down. But by the end of August, September, we will be done. So just pace yourself accordingly. Okay. I guess. Bye.
Thank you.
Thank you.
Thank you, guys.
Thanks, Abhishek.
Bye bye.